Katherine started off at the University of Toronto as a Bachelor of Science student, and then moved to New York University to work with Lila Devachi, and then a postdoc at Columbia with Daphne Shohami. And then she went to UT and started as an assistant professor, where she is now. She's just in that year that you go up for 10 years.
Be kind.
So I invited Katherine after I saw her talk at Love. And one of the things that struck me about that talk is I've gone to many talks about the hippocampus. I've gone to the University of Toronto, spent a whole day listening to people talk about the hippocampus. And I thought I knew a lot about the hippocampus, but I really didn't, I didn't really know what all those regions did and how the hippocampus actually operated. And it was during her talk that I kind of had it like, oh, I get something now. Like all these disparate pieces of information fit together somehow. So I thought it would be great to have her come, and maybe some of you will get that same kind of aha experience. Not too high a bar to set.
Yeah, thank you.
So with that, everybody, welcome Katherine Duncan.
Thanks so much. And thank you so much, genuinely, not just for setting the high bar there, but also for the invitation to be here today. It's really been such a treat getting to meet with the graduate students and hear your amazing questions. I worry some of what I'm covering in my talk will be a little boring for some of you now who've been reading up on this material, but also the great lunch and conversation there. Anyways, I've been enjoying the whole day. And I hope that I can now pay it forward to you by giving you a talk that you can enjoy and that you can gain something from. So I'm not promising that all of you will walk out of this room saying, yes, now I finally get what the hippocampus does, but I hope that you will at least get a perspective on how, as someone who does research in humans, can start to tackle some of these mechanistic questions involving how the hippocampus might form and retrieve memories. So with all of that being promised, something that I really like to start my talks with, especially for those who don't think about memory too much, is this really humbling question of what it is that you're actually going to remember about this talk. So you might have these moments during the talk where things make sense, or you feel like a finding's really exciting and it might stick with you over the long run. And I'm sure all of you have had those feelings in other colloquium which have been delivered in this room. And now if you try to think back on each of those exciting findings and what exactly was being said in those colloquium, it's like a really great memory test, right? Like it gives you a sense of what you actually take away from an experience an hour from then, a week from then. I'm trying to do this with a colloquium a year ago. And you get to these really humbling sense of what it is that your memory is able to keep over the long run, along with this like frustrating question of why it is that you remember some of these things and not other things. What is it that makes you successfully form a memory and other times just fail to have that information stick? And that really is what still remarkably is at the core of most memory research. We've all been attacking this question from a lot of different angles. And something that cognitive neuroscientists like myself often do in order to get some insight into why you remember some things and not other things is to take what could be a really complex experience, but whittle it down to just some core essence. In our case, often the core essence may be an image or a word or images of different types. And this way we can have these really controlled stimuli that we present our participants with one at a time. And then we know what their experience, or we hope we know what their experience was for these different units of time. After completing that sort of what we'll call an encoding or learning session, we can then go through and test our participants' memory for each of these stimuli that were presented, identifying which ones they're able to successfully remember and which ones failed to stick over the long run. And using this approach, we can then backsort our data to identify what's happening in the brain or what cognitive manipulations are leading people to more successfully form memories sometimes, but not other times. And this is an approach that I've used and is one that's been super informative and giving us some insights into how the brain and how cognition and the mind support the formation of memories. But one of the challenges in this approach is it does have a pretty big assumption in it. And that assumption is that what's really important in determining what will stick over the long run has to do with the processes that are being evoked by each of these individual stimuli. So it's an evoked process. And this is in some ways at odds with emerging ideas about how the brain works, where it's not necessarily in the same state at every given moment, but instead you could have slow fluctuations in neural processes or in cognitive orientations that are unfolding even before you get that stimulus in question. And from this sort of perspective, it might be that the state that you're in before that apple is presented is going to be every bit as important for why you later forget it as what that apple then evokes when it is presented. And in line with this intuition that memory waxes and wanes across time, I just have a little teaser here for a new finding from one of my graduate students, Lexi Decker, who literally just wanted to quantify the degree to which memory does actually wax and wane across time. And what I'm plotting here for you is the result of correlating how likely people are to successfully form one memory in relation to forming a memory on an adjacent trial. And what you can see here is that in a task that's really similar to what I was just showing you on that toy computer screen where she presents one image after another, she found that people are reliably more likely to successfully form a memory if they did so on the preceding trial or are about to on the next trial. She also asks how long these fluctuations in good and bad memory states last for by looking at the correlation across trials that are separated by increasingly long intervals. And what she finds is that there is this reliable correlation in memory formation out to 5 trials, or that's about 7.5 seconds in her paradigm. So even in these sort of mundane tasks where each image is unrelated to the next in many ways, people do seem to go through these better states and worse states. So it's just an empirical demonstration of something that I think all of you already have an intuition for. But what I really want to get at is this bigger question of why is it that we go in between these states that are better or worse for memory formation? Why can't we just take it all in? Why won't it all just stick? That's what we feel we want to do a lot of the time. And I think there's lots of ways to tackle this sort of question, and there's probably lots of answers to this question. Maybe it's actually adaptive for us not to remember everything. Maybe we'd be pretty useless if we couldn't prioritize some memories over others. And maybe there's some hard limitations in how much we can attend to across time or how much we can take in a given moment. But what I'm going to talk about today is what I think is a pretty perhaps a more evocative argument that these times where it seems like we're off, it seems like we're not good at forming memories, that these might in fact actually be the times that we're actually good at other phases of memory. So to give you a sense of this, we need to talk about something that makes memory, I think, both really interesting and hard to study in comparison to other sorts of cognitive or perceptual processes that really play out in the here and now, which is that when you're trying to understand memory and understand memory success, it's both a function of how you form that memory, how you then consolidate that memory and reconsolidate that memory. And then lastly, how you then interact with a potential cue that could trigger the retrieval of that memory. And this, of course, is not a new thing. I'm sure all of you have been exposed to these multiple phases of memory before, but I do want to make it really explicit today because part of what I'll be arguing for is that at least some of the processes that could make you good at forming a memory versus make you good at retrieving a memory might actually be at odds with each other. And what that then would mean is that you could perhaps have these trade-offs between the different phases of a memory. And what I'll do is talk about 3 different ways in which you might have competition between processes that would be good for forming memories or learning as compared to processes that would be good for retrieving memories. And the first has to do with your really your attentional orientation or the sort of information that you're focusing on processing. So right now, if you're trying to learn about what it is that I'm telling you, I should hope that you're orienting your attention outwards and taking in the information that I'm providing you, rather than focusing your attention inwards to, remembering what you did for lunch or planning what you might want to do tonight. Which is probably going to make you not very good at forming a memory, but is exactly the kind of attentional orientation that you need if you want to really elaborate on and fully recall a past experience. A second potential trade-off has to do with plasticity. In particular, how is it that some network of neurons is able to take a single experience? And I tend to focus on episodic memory, memories that are formed after just a single exposure. So how is it that neurons can adjust the strength of their connections after the single exposure to be able to encode a particular experience? And that requires a high level of synaptic plasticity to be able to form those memories so rapidly. But in contrast, and when you're retrieving a memory, if you have that same level of synaptic plasticity, that could potentially lead to problems in terms of the overwriting of that memory that you're trying to retrieve and potential interference where you're taking the current experience and interweaving it with that previously stored memory. This isn't to say that that doesn't happen sometimes, but it is a trade-off in terms of processes that could be good or bad for memory formation and retrieval. The last trade-off that I hope to get to in my talk today is one about how we're actually representing the content that we're attending to and that we're processing. And this trade-off has to do with how you deal with basically the level of overlap or similarity that you have across experiences. And so unless you have this extraordinarily exciting life, which if you do, I'm all the power to you, where you constantly have new people and new things you're doing and new places, you probably, most of your experiences are made up of like maybe the people who are sitting beside you right now in this building doing some similar things. So how is it that you're able to actually take those individual experiences and keep them separate enough from each other to ever hope to be able to recall one particular evening over another evening that you had with the same group of people? And this is one case where actually emphasizing the distinctiveness of experiences could be really useful when it comes to reducing interference at the time of memory formation. But by contrast, when you're trying to retrieve a memory, that in a sense depends on the similarity between the cue in front of you and that prior representation, where you need to use the cue to actually reactivate, to make your mind and your brain look more like how it looked when you had that original experience. So all of these trade-offs that I've highlighted could potentially not be a big problem if these processes are being mediated by separate brain regions, separate networks of neurons. And to some degree, that might be true, but there is some pretty good evidence that in particular, this brain region highlighted in red here on my own brain, because I like to show off, is the hippocampus here. That it supports both the encoding and at least the retrieval of recently formed memories. And in particular, the way that we often think about this is that the very same neurons that are involved in encoding a memory are then reactivated. That memory trace or engram is reactivated at the time of retrieval. So when you're thinking about processing in this region or in these networks of neurons, how is it that that can deal with these potential trade-offs? And one long-standing potential solution is that the way that the hippocampus can deal with it is not by trying not to do everything at once, basically. Shifting between operating in states or at least having biases that can separate the encoding and retrieval processes across time. And there's many different models that have made similar claims in this respect, which I'll sort of lump together under this umbrella term of comparator models. There's a few features that they share at their core. One is that the detection of novelty or an expectancy violation would bias the hippocampus then towards a state that primes it for forming new memories when there's this important learning opportunity. But by contrast, if you're in a familiar environment where everything's going according to plan and expectation, the hippocampus would then be in a state where it continues to retrieve memories and make new predictions about what should be happening next. Another common feature across these models is that this bias is mediated through the release of a neurochemical, some sort of neuromodulator such as acetylcholine or dopamine or norepinephrine, where the innervation of that neurochemical in the hippocampus is increased following the detection of novelty and decreased following the detection of familiarity. And it's the presence of this neurochemical that could bias the system to operate in ways that favor encoding versus retrieval. So today I'm going to talk about three different lines of research that fall into these three different trade-offs, each trying to tackle different aspects of this comparator framework. And also each using somewhat different methods to get into these different mechanisms. So I'll start off with a pair of MRI studies where we first begin by asking whether or not the hippocampus is actually situated or produces a signal that reflects this detection of novelty, which then could go on to evoke different processing states. And then returning to this orientation question, asking whether novelty detection can actually bias processing across different hippocampal pathways. I'll then turn to different neurochemical mechanisms that could instantiate these states, starting with an fMRI study and then a pharmacological study that more directly gets at the question of whether dopamine is specifically facilitating the formation but not the retrieval of episodic memories. And then hopefully, if there's time, I'll move on to talking about what I will refer to as putative cholinergic memory states. And I really want to emphasize the word putative here. So this is actually a series of behavioral studies where we've taken what we can find in research performed in non-human animals in order to generate new predictions about how memory would work based off of the effect of acetylcholine in hippocampal networks. And we use this framework to test new predictions, such as whether novelty can enhance pattern separation, which will be a process I'll describe in a few moments that it really emphasizes the distinctiveness of representations. Where on the flip side, whether familiarity can enhance the overlap between representations via a pattern completion process. So now I'm turning to this first study that investigates whether the hippocampus indeed produces a signal that could lead to the internal regulation of these different states. And to get a sense of how this might work, I'm going to start building up a little toy model of the hippocampus here. And I'll also emphasize the word toy here, where I'll be putting in just a few subfields at a time to get a sense of how they might contribute to this sort of mechanism. And the first one that I'll be introducing is the CA1 subfield of the hippocampus. Why this region is so important for this question of expectancy violation or novelty detection is that this region receives input from both area CA3, which has these really strong interconnections between its neurons, recurrent collaterals, that are thought to be important in reactivating memories or generating predictions about what might happen next. Additionally, this region, area CA1, receives strong input from the entorhinal cortex. And this input is thought to reflect what is actually happening in the external world around us. So this positions the subregion of the hippocampus to be able to compare or detect when these internal predictions mismatch with the external reality of what's happening. So in our first study that I'll talk about today, what we wanted to do was to test in humans, is it the case that this subfield is more closely reflecting the degree to which the environment matches or mismatches expectations as compared to other hippocampal subfields? And to answer this question, what we first did was we brought in participants and had them learn through a relentless series of memory tests lasting several hours to be able to um, basically come up with particular room images based off of just the name of the room. So they learned these really rich memory representations of rooms that contain different pieces of furniture and were each given names. And what that allowed us to do was to then bring them back on the second day. And while they were undergoing an fMRI scan, present them with these names of the rooms to first establish an expectation of what they should see next, and then present them with an image. On many of these trials, the image could be the same image as the one that they had studied very well. But on other trials, we introduced changes to these images so that, I'll go back and forth between it there, so that we could manipulate the degree to which the image matches their expectation of what they should see next. We then looked throughout the hippocampus to identify which subregion produces a signal that tracks the total number of changes that were presented in these images. So this involved manually segmenting the hippocampus into 3 regions of interest. One is this critical CA1 region, and another I'll just note is a combined region including the dentate gyrus, area CA3, and area CA2, because there's not good anatomical markers in humans on MRI images to be able to pull those regions apart from each other. And so after manually segmenting these regions, we then searched to identify clusters of voxels that show this parametric response to novelty, and found that the only cluster was in area CA1 with the bold response here plotted to the side, showing how it parametrically increases with the total number of changes in the image. Consistent with this hypothesis that area CA1 could be the region of the hippocampus that tracks the total number of changes in an image. We performed several additional analyses in this study. One to directly compare the signal in area CA1 to other hippocampal subfields to show that area CA1 does in fact track these changes more linearly than other regions of the hippocampus. And we also included a manipulation where we made some changes to the images task relevant and other changes not task relevant to determine whether the signal is produced more automatically or whether it just reflects changes that are important in the moment. And the region that I showed you earlier as best characterized as this automatic tracker of changes regardless of their behavioral relevance. So these results are, again, consistent with the idea that area CA1 is positioned to detect changes. But remember, our big question here is whether these novelty signals, novelty detection, could actually trigger an encoding state in the brain, could prepare it to be forming new memories. So in order to look at that question, I'm going to bring back our first trade-off here, the orientation towards processing internally generated information versus externally driven information. And for those of you who've been paying close attention, first, good work. And second, you might have noticed that there's some parallels here between this distinction that I've laid out and the different pathways that I've highlighted going into area CA1. Where the pathway from CA3 is thought to more so reflect this internally generated expectation of what might happen next, and the entorhinal cortex, by contrast, carrying information from the external world. And indeed, many models have posited that the relative strength between these two pathways could bias the hippocampus towards either forming memories versus retrieving memories. So this is the next question that we addressed, is whether the novelty in these images can actually lead to an increase in the strength of functional connectivity as measured with fMRI between the entorhinal cortex and area CA1, while also decreasing the strength of functional connectivity between area CA3 and CA1. So to test this question, we use the same task here where there are parametric changes, parametric manipulations to the total number of changes in an image. And then what's now plotted in the y-axis is the strength of functional connectivity between the entorhinal cortex and area CA1, where on the right, I'm now plotting the functional connectivity between the dentate gyrus CA3 combined region and area CA1. And what you can see is that with introducing even just a few changes, you see an increase in the functional connectivity along the entorhinal CA1 pathway. But then as these changes continue to increase, you start to see a decrease in the functional connectivity between the dentate gyrus CA3 region and area CA1. So here, these are just combined together into one index where lower scores reflect greater functional connectivity between the CA3 region and area CA1, and higher values reflect greater functional coupling between the entorhinal cortex and area CA1. And this nicely showcases how there's a systematic shift in the relative strength of functional connectivity between these two pathways, consistent with the idea that novelty actually has the power to shift how information might be processed along this circuit. So these results, again, are consistent with the prediction of comparator models that novelty elicits mnemonic states that favor processing of externally driven signals. And I also want to note here, just to bring it back to memory formation and retrieval, in other studies, I've looked at whether you see stronger functional connectivity along these different pathways while people are forming many memories versus retrieving memories, and importantly, have related the strength of functional connectivity to performance on these trials. So that functional connectivity between CA3 and CA1 increases during retrieval tasks as compared to encoding tasks. And it's actually related to how successfully people retrieve memories during those tasks as well. So now returning to this outline and turning to a new trade-off, one that involves plasticity, which basically comes down to how long these memories are going to stick around for. And in particular, I'm going to add this another key ingredient to our toy model now, which is dopamine. And the reason that I'm adding dopamine here is that dopamine, or the presence of dopamine at the time of either LTP induction or memory formation, has been shown to be important in the persistence of that memory or the persistence of that LTP in area CA1. Where specifically dopamine has been shown to support the formation of memories that last over the long term. So long term here, I mean beyond the first phase, the early phase of LTP, and into the later phase of LTP. So at least over the course of 40 minutes to an hour or so. So what we did to build off of this research, which had been conducted primarily in rodent models, was to try to understand whether dopamine could play a similar role in human memory. we started out with an fMRI study to tackle this question, and then afterwards I'll present you with a pharmacological manipulation to more directly get at the dopaminergic mechanism. For the fMRI study, what we did was we interleaved extended periods of memory formation with memory retrieval. Where during the formation or encoding blocks, we presented people with pairs of images, and we asked them to associate those images with each other so that during a retrieval session, we could present just one of the images and ask them to report the image that went with it. The trick here is that the image that they studied on the right was always from a set of 4 repeating images, so that made it easy for them to report which image went with it while they were in the scanner. So in this trial, they should say it was the scissors, not the shamrock that was paired with the ribbon. We then brought our participants back 24 hours later to test the durability of the memories that they formed during this fMRI session. And this was critical because, again, our prediction was that, in this case, functional connectivity between the dopaminergic midbrain and area CA1 would be specifically related to people's formation of memories that last up to that 24-hour period, but not related to their formation of memories that are tested after just a few minutes, like what we did during the scanning session. So what I'm going to plot to you here is the strength of functional connectivity between the ventral tegmental area, dopaminergic midbrain region, along with area CA1. And the strength of that connectivity will be plotted on the x-axis, and on the y-axis, I'm presenting basically memory success. But in this first plot, it's how successful are people at remembering these associations 24 hours later, and based off of the strength of functional connectivity during that initial encoding period. And we saw a reliable correlation there, but importantly, not a reliable correlation in between the strength of functional connectivity during retrieval and people's ability to retrieve memories. And we also didn't see any relationships between connectivity during encoding and their ability to retrieve memories after only a short delay. So we saw a pretty specific relationship here where VTA-CA1 connectivity predicts the formation of long-lasting memories, but not the retrieval of memories and not the formation of memories that last just over the short term. Again, that approach, though, is fairly indirect. It's about as good as we can do with fMRI, but in my postdoc, I really wanted to get into a more causal manipulation to the dopaminergic system. So there we connected a pharmacological study using d-amphetamine in healthy young adults. And I'm happy to say that the study is now finally complete after a lengthy process. And I'm able to share some new data with you here. So the structure of this task was that our participants came on for 3 separate sessions. During the first session, they came in and they formed some associative memories. So they saw 2 pictures and they needed to learn which ones went with each other. And then we tested their memory for half of those associations after a relatively short delay again, so just a few minutes. We then brought them back the second week, and we then tested how well they could remember memories that were formed the week before. We had them form a new set of memories, and then we tested their memory for half of those associations. Critically, the second week was when we had our drug manipulation. So participants were either assigned in a double-blind manner to either the placebo group or to a d-amphetamine group. And then in the third week, they returned. Again, everyone was off drug here, and they simply retrieved the half of memories that they formed the week before while on the drug manipulation in order to see how that influenced their formation of memories that last over the long run. So again, with this complex design, it actually allows us to tease apart some of the potential influences that dopamine could have on memory formation and retrieval. And looking at this first retrieval session, we can ask how dopamine agonists influence the retrieval, but not the formation of memories that are lasting for the long run. During this test, which is really our critical one, we can ask how dopamine agonists influence the encoding of memories that last over the long run. And then during this more short-term assessment, we can ask how dopamine agonists influence memories that are tested after a shorter delay within that early LTP window. Now to walk you through the results of the study. And what I'm showing here is the memory accuracy of our participants on that delayed memory test based off of whether they were retrieving memories on drug or off drug. And you can see that the presence of drug doesn't really influence how successfully people can retrieve associations from their long-term memories. By contrast, the drug did have a reliable impact on people's abilities to form memories that lasted over the long run, consistent with dissociations that have been shown in rodent models. And now for our final test to see if this matches with rodent models, we would expect no impact on the immediate test. But lo and behold, we did see that d-amphetamine increased people's ability to form and retrieve memories over the short term. But before concluding that d-amphetamine and that dopamine in humans doesn't have as specific effects as it does in rodent models, we first need to think about the mode of administration here. So in our people, it was hard enough to get ethics approval to be able to give healthy individuals drugs orally. We could not open up their brains and inject them with the dopamine agonist directly into the hippocampus, right? So this is a systemic mode of administration. And that means that the drug should not just be influencing the hippocampus, it also would be influencing many other brain regions, such as prefrontal cortical regions that are likely going to be playing a role in this sort of task. So for example, this type of rich memory formation and retrieval would also depend on your ability to remain vigilant, have attention, have good working memory manipulations of the material that you're working with. But luckily, we had the foresight to know that would be the case likely. So we also included a task interleaved with our primary memory task, which was sort of like a kitchen soup or kitchen sink task, where we sort of pulled together a lot of factors such as vigilance and working memory abilities and filtering, cognitive control. We just made a pretty challenging task to see how, once we covariate that out of performance, what's left, what seems to be more selective to the long-term memory process. And after doing that, what I'm going to show you here is a plot showing the relationship between the dose of d-amphetamine and memory accuracy when tested after just a few minutes. And here you can see that this dose relationship is pretty much non-existent once we covary out the effect of the drug on working memory and attention abilities. But the effect that we see when looking at memory performance after a long delay is actually pretty minimally influenced by the removal of this cofactor. So it seems like the effect of the drug is, when it comes to long-term memory per se, is actually pretty selective to the formation and not the retrieval of long-lasting memories. Now moving on to the final topic of my talk today, which is this potential trade-off between how you're representing input. And in order to motivate this distinction, I'm going to bring in another neurochemical into our toy model, which is acetylcholine. And this neurochemical is thought to be particularly potent in regulating these trade-offs because it not only influences connectivity along these two pathways that I've already brought to your attention, but it also influences the strength of connections, or the effectiveness of connections within the hippocampal area CA3 network, which is thought to, again, be important for mediating the retrieval, reactivation, or pattern completion of memories, as I'm now going to explain it. And just to walk you through this idea of pattern separation versus pattern completion, and now our toy network model here, each circle is meant to be a neuron and each line a connection between the neurons. And you can see that some of those connections are stronger than others. And that's just meant to reflect a pre-existing memory trace which is stored within this network. This, for example, could be reflecting the memory that you formed the last time you were here in this room. And now when you're in this room again, listening to another talk drone on and on, you have a lot of similar input. But one thing that is thought to be really crucial about how the hippocampus could contribute to forming distinctive memories is that that input could get separated or orthogonalized, such that my talk that I'm giving now might be stored in a relatively non-overlapping set of neurons. But by contrast, pattern completion refers to this mechanism through which partial activation of that pre-existing trace could lead to a completion of the trace with activity spreading throughout it. And this is thought to be important for your ability to reactivate the details of those prior experiences. In order to mediate this potential trade-off, then, what's actually going to bias you towards engaging in pattern separation versus completion when given a partially overlapping experience like today's talk, acetylcholine has been proposed to play an important role there. Where high levels in the context of novelty could suppress the strength of connections between these CA3 neurons, making it more likely that an orthogonal set of neurons will be incorporated into this new memory trace. Whereas in familiar contexts, when acetylcholine levels are relatively low, then those connections are free to activate given this partial input and have activity spread back into the previously formed memory trace. So this is exactly the type of idea that we've been testing, and again, this is where the word putative is going to come up, by testing behavioral predictions that fall out of this sort of framework. And the crux of these predictions have to do with the timing of this innervation. So this cholinergic modulation is thought to have a relatively slow time course lasting on the order of a few seconds. So what that suggests is that it wouldn't just be the case that you engage more so in pattern separation when you have a novel stimulus in front of you, but even recent exposure to a novel stimulus could tip the scales, making you more likely to engage in pattern separation as compared to pattern completion. And that's exactly the type of hypothesis that we've been testing using a pretty straightforward behavioral paradigm where we present people with a series of trials that might just look like standard retrieval decisions to them. But unbeknownst to them, we've actually ensured that there's sequences of new images being presented along with familiar old images being presented. And we position these right before the presentation of critical probe trials that we've designed to assess tasks that should be more dependent on either pattern completion or pattern separation, with a prediction that recent exposure to novelty would improve people's performance on tasks that depend on pattern separation, whereas recent exposure to familiarity should improve their performance on tasks that depend on pattern completion. I see that I am running a little bit short on time, so what I'm going to do is actually skip over some of the evidence, but I'm happy to talk about it later on for preceding novelty, improving pattern separation, dependent task performance, and instead jump right into the converse prediction that recent exposure to familiarity should improve people's ability to engage in tasks that depend on pattern completion. And this is work that my graduate student, Anuya Patil, has been doing, where she is asking people to recall associations. So she has them study a series of images and associates them with individual words. And then during the retrieval task, she represents some of these previously studied images along with new ones. And she asks them to indicate that the image is new if they think they haven't studied it before, to recall the associated word if they believed that they had studied it before. So this would be an associative retrieval task that would depend on completing that initially stored memory that includes the word information. Or they could simply indicate that it's old or familiar if they're unable to actually complete that initially stored memory. She then carefully manipulates whether these judgments follow the presentation of a new or an unrelated old stimulus. And what I mean by unrelated here is that the stimulus was always studied from a different block of time. It was always a different stimulus category. And we never analyzed trials where that was associated with the same word. And so she asked whether just seeing something familiar can actually put you in a state that makes you better at recalling associations. And that's exactly what I'm going to be plotting here on the first graph, where on the x-axis, we're breaking up trials according to whether the preceding image was judged to be new or old. And on the y-axis is how accurate they are at recalling that association. And we found a really robust effect here. So it ranges between 10 and 15% based off of the paradigm. And we see it in over 80% of our participants across our tasks. So this is a super subtle manipulation, but actually a really reliable bias that we can observe, where familiarity improves people's ability to retrieve associations consistent with this pattern completion bias. We asked whether recent familiarity just makes you kind of good at everything by looking at also their ability just to recognize images as being familiar as compared to being new. And she found that by contrast, this item memory that should be less pattern completion dependent does not, is really not influenced by this type of manipulation. Anuya has also varied the amount of time that elapses in between the presentation of her trials to measure the time course of this manipulation, finding that it does last for a few seconds, as we would predict based off of our cholinergic framework. And most recently, she's been conducting an MRI study to try to get at this pattern completion process more directly. So what she does in the study is it has a pretty similar structure where people are learning to associate two things together. They then try to recall one of those things while they're in the scanner. And in this case, they're recalling either famous people or famous places. And then what we can do to index pattern completion using fMRI is to ask the degree to which the pattern of activity evoked when they're just looking at a word but trying to recall Ryan Gosling, does that look less like the other stimulus category and more like the same stimulus category patterns that we observe in an independent localizer scan? And right now, this is still very preliminary data. She's only about a third of her way through data collection, but we are already seeing evidence for greater pattern recapitulation on the preceding old trials plotted there in dark blue as compared to the preceding new trials. So to sort of skip forward a little bit to reach my main conclusion, since it's 3:20 now. This is my ending minute, correct?
If anybody has to go to class, they should go. But
if anyone wants to. Okay. Well, then I'll do a summary before hitting conclusions then. Just to quickly summarize some of the results that I've talked to you about, again, bringing back up this toy model of our hippocampal network, as I begin by discussing how the hippocampus may be positioned to detect violations in expectation or novelty, and then how that novelty could in turn then bias the degree to which pathways that are carrying external information versus internally generated information propagate throughout the hippocampus. I then moved on to talk about this idea of a trade-off between plasticity, where neurochemicals like dopamine that have been shown to be important for establishing long-lasting LTP would be particularly important for the formation of memories, but not necessarily the retrieval of memories. And then in the final study, I showed you just data that's relevant to the similarity end of this prediction, what recent familiarity, which should, in theory, lead to lower cholinergic states, facilitates people's ability to recall the associations that were previously formed. And I had to skip over, unfortunately, some complementary data, and I'm happy to talk about it during the question period, about how recent novelty also has benefits for memory, but it specifically benefits memory judgments that involve emphasizing the distinctiveness. So noticing subtle changes, and we also now have some data showing that it's important for the reduction of proactive interference when forming memories. But then to finish off with some take-home thoughts for you, which as a memory researcher, I know that you can really just take home one thought, but I'm going to say it in 3 different ways to try to make sure that it gets in there. So the first thing to emphasize is that memory processes occur within ongoing neurochemical states. And this feels really obvious, but it's something that, especially as someone who researches humans and spends a lot of time with human researchers, we often sort of like to ignore. And we kind of need to ignore it in some ways because it's hard to get at in humans in the same ways that you can get at it in animal models. And my motto is, I guess if it's hard, try to do it. But not, my motto should be, if it's hard, it doesn't mean that you shouldn't try to do it. And even if our approaches need to be somewhat more indirect, it is, I believe, and I hope that this talk at least got you thinking about how it might be an important factor that can have consequences for complex human memories. So when we're thinking about these sorts of episodic memories that rely on all of these constituent processes, it's really important, I would argue, to start thinking about all of the underlying mechanisms that are responsible for these constituent processes and how they might fluctuate on the basis of these constantly changing neurochemical states that exist in our brains. And then lastly, something that I really like to emphasize is that by focusing in on this comparator, this novelty or expectancy violation mechanism as an organizing principle for establishing memory states, what it does is it actually highlights the potential adaptive consequences of these states. So it's not that we're just aimlessly wandering around with brain states that might match what we need to do or might not match what we need to do, and you get lucky sometimes and you don't get lucky other times. Sure, there's going to be part of that at play, but it seems like the factors that could establish these memory states are exactly the sort of factors that you would want to have in place so that you could adaptively shift in and out of different biases or modes at the right time. And one reason why this is so important to get a handle on, really the adaptive regulatory role of neurochemicals in memory, is that so many disorders of memory have a neurochemical underpinning. And rather than thinking of these disorders as either being disorders of memory as this unitary construct, but starting to dig into how these neurochemicals or the dysregulation in them is actually leading to a lack of adaptive memory, a lack of fine-tuning of memory operations to occur at the right time. I think we can start to get some better insights potentially into the nature of these disorders, which is a direction that some of my graduate students are now starting to go down. So with that, thank you so much for the invitation to be here, for coming and sitting through the whole talk. And also, of course, a big thank you to my graduate students, in particular, Anuya and Lexi, who are up top and contributed to some of the research that I discussed today, along with my former mentors, Leila Davachi and Daphna Shahami, as I presented many of the ongoing collaborations and work that I had done under their mentorship. Thank you.
So if anybody has to go to class, Andrew?
Thanks for a really thoughtful talk, really interesting stuff. So I guess one question I had is, I was thinking about the interplay. Like you kind of described it as kind of like a retrieval state, state.
Yeah.
And I guess we toss around those words a lot. Retrieval, and they're very useful terms. But on one hand, I'm thinking about the interplay between for example, it's one thing for you to say, I want to encode your talk really well, right? But maybe it sparks an idea from a different lecture in this exact room from whatever 2 weeks, 3 weeks ago. So in that case, part of me wants to pattern complete, but then shortly after to kind of encode separately the things that you're talking about. So I guess I'm wondering about how these things might, in your mind, how they interplay with.
Yeah, I think that's a great question. And I think one of the benefits of, I know a lot of my talk was organized around encoding and retrieval, but what I'm hoping to do is to start to get into the component neural processes that go into encoding and retrieval. And I think once you can start doing that, you can start thinking about things like, integrative encoding, for example, which is what you're describing there. So, in the simplest way, it seems as though it's best to be taking in what's happening around you if you want to form a veridical memory of what's happening around you. But sometimes for what our lives require, getting the exact details of what I'm saying might be less important than connecting it to prior knowledge so that you have the ability to later at the time of retrieval be able to access this information when you're thinking about related information. And then that now puts you in a place where you might want to have high plasticity, but have pattern completion happening at the time of this high plasticity, which so that would involve forming and changing the structure of your memory while not engaging in pattern separation. And these are, sort of once you start having these ingredients, you can start thinking about the trade-offs between these different components and also start thinking about, why is it that we have all of these different neurochemicals that might be adjusting these trade-offs in slightly different ways. So something that I've argued for, but I think there needs to be a whole lot more evidence for, is that, for example, being in a higher dopaminergic state but a lower cholinergic state might be particularly helpful for exactly those sorts of circumstances where you need to not be engaging in too much pattern separation. You don't want that much inhibition in your hippocampal networks, but at the same time, you want to have high plasticity in order to incorporate this new information in with old information. So I think that's a great question and exactly the sort of line where you always, I think, need to start with the simplest way of thinking. And then once you've obtained some evidence for those sorts of dissociation, then start to add in the nuance to think about these different combinations of factors and how they could play with each other.
Thanks.
Yes?
Sorry, this is a little bit related. One of the older ideas that's similar to what you're talking about, but at a very finer time scale, is that you alternate between encoding and retrieval within a
Theta. Yeah. Yep.
So Mike Hasselmo
Yeah.
If I remember correctly, he was also suggesting it was cholinergic
Yep.
modulation.
Although the timing didn't work out for that, so he then ended up revising the. So I am clearly a big fan of some of his earlier work, thinking about cholinergic regulation of these states, and then the time scale ended up not matching up because a lot of the cholinergic effects were too slow to be able to be fluctuating at the theta phase time scale. But something that is really interesting is that there is, of course, strong relationships between acetylcholine and theta, where higher cholinergic states are associated with stronger theta oscillations, and lower cholinergic states are associated with higher sharp wave ripple frequency. Which is, again, very consistent with this idea of having more encoding versus retrieval modes, with the sharp wave ripple periods being important for the pattern completion mechanisms. But then what you end up having is this interesting situation where you could have states embedded in states. So in your high cholinergic state, as you cycle through a theta phase, you might actually still have some shifting within the single theta phase between a state that's more attuned to retrieval versus encoding. And this is exactly some of the work that I'm trying to get into with a collaborator where we can do deep brain stimulation in the hippocampus, actually at different phases of theta, to see if in humans we can replicate these findings about LTP dependence on theta phase in the human brain as well. So that's an ambitious project, one of many, to try to bring in the theta timescale as well. And maybe one other thing I'll mention in relation to that is Maria Wimber's group recently had a really nice study using MEG as well that related the degree to which she could index pattern completion in the cortex with the phase of theta, basically, as identified and source localized to the medial temporal lobe, which is very much consistent with this fast oscillation idea between encoding and retrieval states, which I believe would be sort of superimposed along these longer time scale biases that I've been studying up to this point. Yeah.
Study used division of the sensory input for community. Olfaction is known to be a quick memory trigger. So what do we know about the entorhinal cortex and its input in sensory? Can you measure the activation from different sensory?
Yeah. So olfaction we know so little about in humans, I would say. There's one researcher in my department with an olfactometer, and we've talked about potentially trying to learn more about olfaction. I know we all have this idea that olfaction is such a good memory cue, and in particular, I don't know how much this holds up in the human brain. Definitely in the rodent brain, there's really strong olfactory connections to the medial temporal lobe. I think in the human and even non-human primates, the visual input it's more predominant as compared to the olfactory, though. And I don't know, there must be work out there that I'm unaware of. If anyone is aware of it, please enlighten me. I'm looking at olfactory memory cues in humans, but I don't think we know much when it comes to the cognitive neuroscience of this phenomenon, apart from a few studies that use olfactory cues during sleep as a way to be sort of like priming memories for consolidation during sleep. So olfactory cues are really helpful during sleep because they don't wake people up as much as auditory cues, and they don't require you to pry their eyes open for visual cues. So I think it's a great question to start to understand, does this work differently in other modalities? Something that is much more straightforward to get at, which I'm hoping to incorporate soon, is more auditory cues. So this is a modality that's very straightforward to implement in humans, and would be really interesting to see whether you can get cross-modality effects. So a novel sound, for example, does it have the same effect on visual memory as compared to within modality novelty? What sort of transfer do you have there? Hi.
So your study focused on
Okay. Mm-hmm.
and so if you're trying to get at the plasticity issue, have you looked at any structural imaging?
We have not yet, but that is actually on sort of in the pipeline, as you would say, I suppose. So my newest graduate student is interested in, especially in aging, actually doing studies looking at structural changes longitudinally and how structural changes in the basal forebrain, but in parallel, we'll also be looking at the hippocampus are related to different biases that we see in memory as these individuals age. So we are now starting to get at some structural imaging techniques as well. It's a great suggestion.
Beyond hippocampus, the cortical areas, and I'm thinking about one of the authors that I think were published earlier this year, showing structural changes in precuneus in the visual perceiving task very quickly.
Oh wow, that's impressive. No, so we haven't looked structurally at very much, and then functionally, so the data that I presented, that little teaser data about looking at pattern completion indices as people were performing this associative memory task, that was from the ventral stream. So it was outside of the hippocampus. Generally, you see higher fidelity reinstatement in cortical areas as compared to the hippocampus. So we will be looking directly in the hippocampus, but also across the ventral processing stream with these stimuli. But we additionally have parietal regions that we'll be looking at as well, where in particular the precuneus has been shown to have pretty strong pattern reinstatement during retrieval. So we are looking, I've made a joke over lunch about how I've been sort of kicking and screaming my way out of the hippocampus since starting my lab, and how helpful it is to have graduate students with interests extending outside the hippocampus to start me thinking and start my lab thinking in more of an integrative way across the brain. Yeah.
So I want to think a little bit about event segmentation and how these time courses can tell you something about what's an optimal duration for an event. Because if you constantly have novelty and you're constantly encoding, how long can you keep that state up?
Yeah.
And so is there something there when you think about these time courses that would tell us, like, for a learning bout, what's the...
Yeah. Well, so that's exactly, I gave you that little teaser in the beginning of my graduate student Lexi Decker's work on autocorrelation and memory formation. And in one of her planned studies, so what she has been doing in addition to looking at just autocorrelation in memory is trying to characterize on a trial by trial basis fluctuations in sustained attention. And we've been looking at that and how it relates to memory formation in both healthy young adults and in kids, and characterizing what these fluctuations look like in these different populations. And one thing that we can definitely see is that people have their own rhythms, it seems. So there's wild diversity in how stable people's attention seems to be across time, where some people, her encoding task is like a particularly boring one, but it only lasts for 8 or 10 minutes or so. And some people can really just stay focused and in the zone throughout that period of time. And other people, you just see them wildly fluctuate across time. And something that she's wanting to look into is there like an optimal rhythm? So is there an optimal rhythm for each person? And is there an optimal rhythm overall? And part of this work is inspired by some research that Kei Fukuda, who's at U of T now in the Mississauga campus, has done, looking at just instructions that people are given to like form a good memory now versus this one doesn't really matter. And what he's found is that if you tell people to like really try hard on every trial, that is identical pretty much in terms of memory performance as telling them that nothing matters. So like it does seem to be the case that we can't stay on for the whole time. He's only looked at it at a few different proportions. I think I'm probably wrong here, but 20% and 50% as well. And it seemed like somewhere around 20% seems to be optimal. So people can't necessarily be on for that long, and they can't be on for too much, like too higher proportion of the time, it seems. And he hasn't looked at the like temporal profile of this at all. He just has randomly instructed people to be on task or be off task. But what we're planning on doing with him, with Lexi's work, is to be characterizing these rhythms and give people instructions that match their rhythm or that match like the best rememberer's rhythm, to try to see something more about like the nature of the timing of these things. Definitely from that perspective, we're thinking of it more from like a cortical state rather than a hippocampal state per se. But I do think it's really interesting to now start thinking about, again, this looking beyond the hippocampus, but when these neurochemicals are being modulated by factors such as like novelty, we also use reward and other sorts of salient events, they're not just being modulated in the hippocampus. And there may be some degree of independence in these projections, but it's very likely that if acetylcholine's going up in the hippocampus, it will probably be going up in more posterior regions and in prefrontal regions at the same time. And what does that do to coordinate processing across the brain, really? So is it good to have high cholinergic states throughout, or can there be some downsides of having high cholinergic states in cortex and having low cholinergic states throughout? I'm getting rambling now, but basically, these are some of the questions that we're trying to tackle. Yeah.
I don't know if there's any data on this, but I'm thinking about the upregulated sort of functional connectivity between the frontal cortex and CA1. And the simple question is, how fast after experiencing novelty do we know how quickly you start to see upregulated functional connectivity?
Yeah, that's a great question. I don't know if we have the answer to that question. So with MRI, it's not a particularly good tool for getting at the precise timing there. And in humans, using MEG or EEG obviously would be sort of a disaster if you're trying to tease apart sources in hippocampus versus entorhinal cortex. So that is really a question for more invasive techniques. And at least to my knowledge, I don't think that those parallel studies have actually been done in non-human models. Or
nobody knows what attention is.
Yep.
But we seem to think we know that it's going to novelty. And so I'm just wondering if that increased functional connectivity could be like a mechanistic of what we long meant by novelty. Functionally, there's novelty upregulates encoding. And of course, when you've got something encoded, you'll then might move your eyes to it, for example, because it has functional existence.
Yeah. And that is, so we are doing some work in MEG now, looking at selective attention to particular stimuli to see how the selected stimulus content might like propagate across cortical regions on its way to the hippocampus, like sort of selective filtering of information. And I think that will give us some greater timescale resolution to try to get a sense of, when you present like this novel face, how does that event unfold? And we won't be able to have the same sort of spatial resolution with it, but it would give us a better intuition, definitely, of the sort of the staging in these shifts.
Okay, so before we thank Katherine, there's a wine and cheese up on the second floor. We'll meet you there. And thank you very much.
Hey, thank you. Oh.